All right, my friends, we'll get started. Welcome back. Hope you had a good weekend. So we're going to finish with phospholipids today, fat-soluble vitamins, and prostaglandins. We will get to terpenes before the end of class, but terpenes will not be on this exam. Did I press record? Yeah. As we talked last time, phospholipids and sphingomyelins have the same basic character of polar head group with two nonpolar tails. You can recognize a sphingomyelin as different from a phospholipid because phospholipids have esters, 3-carbon glycerol unit, 2 esters with fatty acids, and then a phosphate-linked polar head group. We talked about how these polar head groups can be negatively charged or neutral. Zwitterionic, meaning charged but neutral overall. Sphingomyelins are a little bit of a different beast. They still have 2 nonpolar tails and a polar head group, but the one fatty acid in a sphingomyelin is linked via an amide bond. And then the other nonpolar tail is sort of in the same carbon chain as the phosphate head group. These aren't structures really to memorize, but you should recognize the difference between them. You should be able to pick them out of a lineup, which is a sphingomyelin. And again, that's easy. Just look for the nitrogen and look for the amide bond. Okay, so I think towards the end of class last time, as we talked about phospholipids, and sphingomyelins, I said that the 2 nonpolar tails give the phospholipids sort of a cylindrical profile, whereas a fatty acid or other detergent has 1 nonpolar tail and a polar head group. It has more of a conical shape. By the way, fatty acids are not the only things that form micelles. Many detergents have the same basic structure of a nonpolar tail, but will have a different kind of polar head group. There are a lot of different polar head groups that are involved in detergents and cleaning products, but most of them will have this conical shaped profile. When they associate with each other side to side to bury their nonpolar surface area away from water, the conical shape gives that assembly substantial curvature. And so you end up forming spheres that have a nonpolar interior and a polar exterior that interacts with water. And this is why soap is good at removing grease and oil, because the oil and grease is soluble in the interior portion of the micelle, whereas the micelle overall is totally soluble in water. So this is how soap works, and originally our ancestors made soap by hydrolyzing triglycerides to form fatty acids. A lot of soaps, detergents, shampoos are no longer fatty acids, but they still have a nonpolar tail and a polar head group. Making the phospholipid or the sphingomyelin have this, the 2 nonpolar tails give it a cylindrical profile, side to side self-association, to sequester the nonpolar surface area from water, doesn't have a lot of curvature, and so if, depending on concentration, you may only have, of the phospholipid, you may only form a monolayer. When you form a monolayer, you just have the polar head groups interacting with water, with the nonpolar groups sticking out into air, because it's better for them to be exposed to air than to be exposed to water, where they'll experience the hydrophobic effect that we've talked so much about. As concentration increases, though, 2 monolayers can associate with each other with their nonpolar tails in the middle as sort of a nonpolar tail sandwich between 2 strings of polar head groups. Then either side of the bilayer is fine interacting with water. And this bilayer can extend indefinitely in left and right, forward and back 2 dimensions to create this sort of sheet of polar exterior and nonpolar interior. And over time, that will curve back on itself to form a larger assembly Now, distinct from a micelle, in the micelle, the polar head groups were on the outside, the nonpolar groups were in the interior. In a bilayer, you have polar head group on the outside, nonpolar, intermediate, and then polar group on the inside. You have an outside water area, an inside water area. This is on a small scale called a vesicle, but as the molecule, as a vesicle gets bigger, you can get to things that are like cells. And the compartments inside the cell, nucleus, ER, mitochondria, there's membranes associated with all of these. And so bilayers are nature's reaction vessels. They're ways to make different parts of the cell that have different environments. Sphingomyelins are incorporated into lipid bilayers, and different types of cell membranes will have different amounts of sphingomyelins. As we said last time, the myelin sheath surrounding neurons is particularly rich in sphingomyelins. The other thing that you'll learn more about in biochemistry is the properties of the bilayer depend on the fatty acids that are in the phospholipids, which we haven't said a lot about. Some of these can be saturated, some of these can be unsaturated. Phospholipids that have unsaturated fatty acid chains are going to have cis double bonds in them. And you can imagine that a fatty acid with a cis double bond, which I'm going to just illustrate here briefly, might have different assembly properties than a fatty acid without a cis double bond. The fatty acid with the cis double bond is going to have a different shape, and that might not associate as tightly with fatty acids that don't have double bonds in them. The composition of phospholipids can change what's called membrane fluidity. Membrane fluidity is how fast does one phospholipid diffuse through the membrane and walk through this two-dimensional sheet from one place to another. You might imagine that as a membrane has more unsaturated fatty acids in it, that the individual phospholipids don't pack quite as tightly, and so maybe the membrane is more fluid. You'll learn more about this in biochemistry. A lot of things get embedded in membranes. There are membrane proteins that have nonpolar portions that span the bilayer, and then polar portions that occupy internal and external surfaces of the protein. A lot of these of the bilayer, a lot of these membrane proteins are involved in signaling from outside the cell to inside the cell. Okay, so that's phospholipids. What we want to do now is talk about, we're going to spend the rest of our time looking at the structures of and talking about vitamins that are basically lipid soluble. They can accumulate in fat tissue, and then also prostaglandins and eicosanoids, which are involved in pain and inflammation and some other things. So first, fat-soluble vitamins. There are some vitamins that are water-soluble. If you eat too much vitamin C, for example, nothing happens to you because your kidneys filter out the excess vitamin C and you just pee it out. And as somebody on Big Bang Theory once said, when you take too much vitamins, you're just making very expensive urine, so knock yourself out. For the fat-soluble vitamins, it's not that way because these vitamins are not that soluble in water, so they tend to accumulate. So you can accumulate too much of these, though generally with the American diet, that's not a problem, right? It's not like we're getting too much vitamins. Anyway, so these vitamins are named with letters. The letters don't mean anything. There's some historical context to why one is A versus B versus... There are many B vitamins. There's only one vitamin C and one vitamin A. There's not a lot of rhyme or reason. The B vitamins don't all do the same thing. And so it's kind of confusing, but we'll navigate it as best we can. We're going to learn about 4 of them. The easiest is vitamin A because you've seen molecules like this before. When we talked about vision, we talked about a molecule called 11-cis-retinal. Your body actually makes 11-cis-retinal from vitamin A as a precursor. So vitamin A has a 6-membered ring, and then it's fully conjugated, and it's got a long string of adjacent atoms with double bonds, and then there are branching methyl groups at regular intervals. So this is vitamin A. Another word for it is retinol. And again, it's a precursor to 11-cis-retinal, which gets loaded onto your rhodopsin protein in your eyes as an imine, and then that absorbs light and isomerizes, and that allows you to see. Retinol is also involved in skin health. So if you have a vitamin A deficiency, you'll have vision problems, especially night blindness, but you can also get dry eyes and skin. Retinol itself is important for healthy mucous membranes, which, nose, mouth, epithelial cells, and so on. Okay, so that's vitamin A. Vitamin A is characteristic of a class of molecules that we'll talk about next time called terpenes. One of the ways to recognize a terpene is there's a dimethyl, 2 methyls attached to the same carbon, and then there's regular spacing of branching methyl groups in the molecule. And that's a dead giveaway for a class of molecules called terpenes, which we'll learn about and learn how to synthesize them. Okay, you can see that it's mostly nonpolar, which is why it's called fat soluble. The next one is vitamin D3. So this is synthesized from a variant of cholesterol. And you also need, interestingly, UV light to make it happen. And so you'll hear people say, oh, I really need to get outside, I need more vitamin D. And the reason is you need some exposure to UV light in order to make the reaction that generates vitamin D happen. We maybe will get to that reaction this semester. It's a photochemical reaction. It's kind of cool, but I've never yet gotten to it in the past 5 years, so maybe we'll be the first. I'll show you the structure of vitamin D. It's got a 6-membered ring and a 5-membered ring fused together. When you learn the structure of cholesterol, you'll see the similarities. There's a portion of the molecule that branches off from the 5-membered ring. And if you're looking closely, there is the dimethyl, sort of 2 methyls on the same carbon, what I like to call a dimethyl tail. That's characteristic of terpenes, and ultimately cholesterol is synthesized, is an example of a terpene molecule. You can see that it's mostly nonpolar. You have 3 conjugated double bonds, and those are the remnants. I'll just highlight them briefly. 1, 2, 3, 4, 5, 6. Those are the remnants of what used to be a 6-membered ring that opened up. And, well, it's not exactly that way. I'll unhighlight it because what I told you was only true from a certain point of view, and I'm not feeling like Obi-Wan today. There's actually 2 reactions that happen. There's a ring opening, and then there's a rearranging of the double bonds. It's called a sigmatropic shift. Anyway, like a lot of these, you have an OH group and then a nonpolar tail. Vitamin D3 is involved in regulating calcium and phosphorus metabolism. Both of those minerals are important for bone growth and maintenance. So if you have a vitamin D deficiency, and as you're growing, you can end up with a condition called rickets, which involves bone deformities and spinal curvature. This used to be really common in the United States, and this is why vitamin D is in milk and added as a supplement to milk, just to prevent this from happening. Also an interesting story is, as I said, you need UV light to make this. And so, but we know that UV light can also cause problems. There's another photochemical reaction that can occur in your DNA where UV light causes two thymine bases to interact with each other and form an unnatural bond. And if you get enough of that DNA damage over time, you can get cancer. And so humans have to navigate this sweet spot between getting too much sunlight and too little sunlight. Too much and you're going to get sunburn and get skin cancer. Too little and you're not going to have enough vitamin D. So it's kind of an interesting balance. Okay, let's now do vitamin E. Vitamin E is an antioxidant, and its chief function is to protect double bonds in fatty acids. So just as a quick reminder, I'm going to scroll back to a previous lecture. Last time we talked about what can happen to double bonds in fatty acids. And that is that the allylic carbons are especially prone to oxidation. Oxygen, molecular oxygen, is like a diradical, and you can abstract a hydrogen atom from the allylic carbon to generate a resonance stabilized radical that gets modified with a peroxide group, and you end up with a damaged fatty acid, right? And that can change its properties, certainly changes its smell and taste. Oxidative, you've heard about reactive oxygen species and oxidative damage to cells, and this is some of what goes on. So what your body needs is something that can intercept the oxygen radical to prevent oxidation of allylic carbons in fatty acids. So you need what's called a sacrificial oxidant, meaning some molecule that's willing to fall on its sword and give up a hydrogen atom and get oxidized. The best choice would be something where it's really easy to remove the hydrogen atom, but where the resulting radical is really quite stable. You might call this a radical trap. And so here is the molecule. It's got a benzene ring with 3 methyl groups on it and an OH called a phenol. Phenols are really good sacrificial oxidants because if you can imagine molecular oxygen removing a hydrogen atom to generate a radical on the oxygen, that resulting radical, and I'm not going to draw the whole molecule, that resulting radical would be resonance stabilized. We won't draw the resonance structures, but hopefully you should be able to see how that radical could be delocalized into the benzene ring. This is, as I said, a resonance stabilized radical, and it is not particularly reactive. It's content to sit on its own for a while, and so it can intercept oxygen and prevent oxidation of allylic carbons in fatty acids. There are nonpolar tails. There's a nonpolar tail to this molecule, and it's got branching methyl groups at regular intervals. So this is also an example of a class of molecules called terpenes. There's the dimethyl tail. And you could imagine this embedding itself in membranes where the phenolic OH group would be ready and waiting to intercept oxygen to prevent damage to fatty acids. If you have a vitamin E deficiency, you can have neurologic problems. And there are a lot of other antioxidants that are similar to vitamin E. You'll hear about molecules that are polyphenols. That's molecules that have lots of these benzene rings with an OH group on them, and they're antioxidants via the same mechanism. They intercept oxygen, give up a hydrogen atom, but are pretty stable and unreactive on their own. All right. The last one is vitamin K. And vitamin K looks similar, really similar to vitamin E, only vitamin K isn't a phenol. So vitamin K has the same sort of benzene ring structure. It has a 6-membered ring fused to the benzene ring, but its structure is totally different. And then it has a nonpolar tail that looks like this and has the same alternating pattern of carbons and then branching methyl groups. For vitamin K, the nonpolar tail is a little bit longer. Vitamin K regulates the synthesis of a molecule of a protein called prothrombin. That THROMB sort of root is related to clotting and platelet aggregation. So prothrombin is a protein that promotes blood clotting. And there are a number of these, and vitamin K promotes the synthesis of prothrombin and other clotting proteins. So if you have a vitamin K deficiency, you can have inadequate blood clotting. All of these, as you can see, are nonpolar. And you can imagine them accumulating in tissues that are rich in triglycerides, and some of them you can imagine being embedded in membranes. All right, questions about the vitamins? Okay, well today, yeah, go ahead.
So, do we need to get these structures down, or what should we
Do you need to get these structures down? I would say not at the level of being able to draw them from memory, because I can't. But at the level of recognizing them, yeah. So, I would work on looking at key features of them that might help you remember them. Vitamin E, the phenolic OH, is really the key, the benzene ring with the OH on it. For vitamin D, I would say this fused 6 and 5-membered ring is the key structural feature. Retinol, I'd say remember retinal in eyes, remember the fully conjugated structure, and that would be the giveaway. And then vitamin K, I would say it looks like vitamin E, but its nonpolar tail is longer. Also, it's a diketone maybe instead of a phenol. Okay, other questions? All right, so our next story is the class of molecules that are called eicosanoids. And they're called eicosanoids because they come from a 20-carbon precursor. And I'm a 20-carbon fatty acid precursor. And I'm not sure, eicosa, is that 20 in Greek or something like that? Something, icosahedron is a 20-sided something or other, okay. So the precursor is something that we talked about last time, arachidonic acid. I'm not sure how to, arachidonic, yeah, something like that, acid. If I spelled it wrong, I'm sorry. No, that's right. Arachidonic acid is a 20-carbon acid with 4 double bonds. So it's highly unsaturated. And the way it's drawn in your text is in a sort of pseudo-symmetrical orientation. One way to remember it is that you have 4 double bonds and they're each separated by an allylic carbon. Notice the double bonds are all not conjugated with each other. They're all cis and they're all separated by allylic carbons. And then on the one side you have 4 more carbons, with the last one being a carboxylic acid. And then on the other side you have 5 more carbons. overall 20. Okay, so that's arachidonic acid. And this is the precursor to a whole bunch of things that are important signaling molecules that regulate pain, inflammation, muscle contraction, gastric secretions. And they're important to know about because there's a lot of drugs that actually target these molecules. So let's talk about a few of these. There's an enzyme called 5-lipoxygenase. You don't need to memorize that enzyme, but that enzyme is involved in making a class of molecules called leukotrienes. So as we draw this structure, see if you can identify the 20 carbon arachidonic acid, and see if you can see why they call it a triene. It was a very triene time. Yes, that's yes, Santa's with us today. All right, so in leukotrienes, the 20 carbon arachidonic acid has been modified. One of the cis double bonds is still there. Actually, two of them are still there, separated by an allylic carbon. But then there are two additional double bonds that aren't in the right place, and they're not cis anymore. So you have these two additional trans double bonds that are conjugated to this cis double bond. That's the triene portion. So if you want to recognize a leukotriene, look for the three conjugated double bonds. Immediately adjacent to those three conjugated double bonds is a sulfur, and then on the adjacent carbon is an oxygen. Then 4 additional carbons, and you have the carboxylic acid. So you can see why the enzyme is called 5-lipoxygenase, because on 4 carbon 5, there's an OH group. Now, we're not talking about the synthesis of leukotrienes, but if you were doing, and there's something else attached to this sulfur, but if you saw an OH group and on an adjacent carbon something that could have been a nucleophile, what would you have done to make this? Epoxide opening, right? That makes sense. It's something you learned in 351. Think in retrosynthesis. Nucleophile opens up, the epoxide ring opens up, nucleophile and OH are trans to each other, and they're on adjacent carbons. Nature actually uses an epoxide to do this. You may or may not learn about that mechanism in biochemistry. So the thing that's attached to this sulfur, the sulfur is actually the side chain sulfur from a cysteine amino acid. So the other portion of a leukotriene is a tripeptide. So here is the cysteine portion, and then the cysteine carboxylic acid is linked via an amide bond to a glycine residue. So this is cysteine with the side chain sulfur bonded to the carboxylic acid. And then the amino group of cysteine is bonded to the side chain carboxylic acid of a glutamate residue, which is kind of weird. Normally we would expect the backbone carboxylic acid of glutamate to be the one involved in amide bonds, but here it's the side chain. So this is a leukotriene. This one's called leukotriene C4. There are a number of these that differ in their structures, differ in what's attached here. Leukotriene C4, leukotrienes in general are involved in smooth muscle contraction, especially in the lungs. So they can play a role in asthma. So one of the challenges of asthma is if your lung smooth muscle constricts too much, it's difficult to inhale. And so regulating leukotriene synthesis is one of the strategies you can take for dealing with asthma. Okay, so do you need to memorize that structure, draw it from memory? No, I can't. If I show it to you and say, which of these is a leukotriene, can you answer it? Probably, and that's actually a pretty easy question as long as you remember to look for the 3 double bonds. All right, let's talk about some others. So notice that in the leukotriene, the 20 carbon chain from arachidonic acid is basically linear. We've just made an epoxide here and then attacked it with a nucleophile. For the others, there's some really strange cyclization that happens. And the story is actually important to medicine, and actually the story involves BYU as well. So this is arachidonic acid, and there are a class of enzymes called cyclooxygenases. They are going to oxidize and cyclize arachidonic acid. And the enzymes that do this, there's one called COX-1, and there's another called COX-2. COX-2 was discovered here at BYU by Professor Dan Simmons, and he is the person for whom the Simmons Cancer Research Center is named. And he taught me biochemistry when I was here as a student a gazillion years ago. He's since retired. He discovered the enzyme, and that discovery led to the development of several drugs, which I'll tell you about in a little bit. So let's show you what these enzymes do. They make, as a first step, a precursor to the rest of the molecules that we're going to talk about. There's a 5-membered ring, and that 5-membered ring comes from these 5 atoms. And then, this is really weird, the 5-membered ring is bridged by a 2-oxygen bridge, which comes from molecular oxygen. So that's where the cyclooxygenase comes from. We made a ring and oxygen's part of it. Another feature that this intermediate has is a trans relationship between 2 tails, which are the remaining carbons of arachidonic acid. In the one on top is essentially unmodified from arachidonic acid. You still have the cis double bond and then the carboxylic acid. The one on the bottom has a trans double bond, and it has an allylic oxygen. There's been apparently some allylic oxygenation, and in the precursor, that's actually a peroxide. That's the same kind of intermediate that we showed happens when fatty acids get oxidized at the allylic carbon. You don't need to memorize this, but if you ever learn more about it, this intermediate, this precursor to a lot of the molecules we'll talk about today is called PGG2. A lot of these molecules are going to have this 5-membered ring structure, and they're going to have these 2 tails. All right, let's keep going to talk about the sort of downstream descendants of these molecules. So if you take PGG2 and you reduce this oxygen-oxygen bond so that it's OHs on both sides, you have an example of a prostaglandin. And the specific one we'll talk about, though there are others, is PGF2 alpha. They have different effects. Here's again the 5-membered ring, and those oxygens that used to be attached to each other are now free, but they're still dashes. You've still got the same tail on top of the carboxylic acid. The difference in the lower tail for PGF2 alpha is that the allylic oxygen is now just a hydroxy group rather than a peroxy group. Okay? Prostaglandins are, and the effects are differ depending on the kind of prostaglandin, but in general these can lower blood pressure, but you're going to think that that's boiling point, so blood pressure, lower your boiling point, that would be kind of bad. This particular prostaglandin can inhibit platelet aggregation. It can lower gastric secretions. And it can regulate or control inflammation. So a lot of prostaglandins and eicosanoids are involved in pain and or inflammation. Another class of molecules, I don't have enough space to do all three of them here, so I'll just have to erase one of those arrows and we'll keep going. Is going to have the exact same tails as PGF2A, but it's going to have a different ring structure. This is called thromboxane. And you can recognize a thromboxane because they have a 6-membered ring where one of the atoms is oxygen. And then they have this other weird oxygen that's a dash at both of these positions. So you have a 5-membered ring and a 4-membered ring fused together. Each ring has an oxygen in it. Other than that, the tails are exactly the same. You have the upper tail with the cis double bond and the carboxylic acid, the lower tail with the trans double bond and the allylic alcohol. Thromboxanes are involved in constricting blood vessels, and you can tell based on that THROMB word in the name, they are related to triggering platelet aggregation. And then lastly, and again, you can recognize a thromboxane with this weird 6-membered ring structure in the head group. All right, the last class of eicosanoid that we'll talk about has the same 5-membered ring structure with the wedge OH on this side. It's also going to have the trans double bond in the lower tail with the dashed allylic alcohol. By the way, I know I said this was a wedge, it's a dash, I'm sorry about that. Up here, it's going to have a dash oxygen and a dash, but the upper tail and that oxygen are actually going to be linked together. Then there's a double bond here, and then the carboxylic acid there. This is called a prostacyclin. Cyclin because it's got an extra ring in it. This is called PGI2. Prostacyclins are involved in dilating blood vessels and inhibiting platelet aggregation. So it's interesting that you have related molecules from the same precursor. Some prostaglandins inhibit platelet aggregation, some thromboxanes promote platelet aggregation, prostacyclins can inhibit platelet aggregation. You have opposite effects with similar structures. Why do you suppose biology needs molecules to both promote and inhibit platelet aggregation? Go ahead.
For blood clotting, like you don't want your blood too thin or too thick.
Right, so there are bad things that happen when blood is too thick versus when blood is too thin. If your blood doesn't clot, what happens? You can bleed out, right? If your blood clots too much, what can happen? You can get heart attacks, stroke, limbs can go cold. A lot of bad things can happen, right? So you need a complicated system that maintains homeostasis, maintains balance, so that you're not too far to one side or the other. Good. Now, there's an interesting story with COX-1 and COX-2, the enzymes that make this precursor. Aspirin and other non-steroidal anti-inflammatory drugs block the cyclooxygenase. So that's the NSAIDs like aspirin or ibuprofen. These actually bind in a tunnel in the enzyme that the nonpolar tail threads itself down. They block the nonpolar tail, and so the synthesis of the prostaglandins can't happen. And that blocks pain. Prostaglandins are not very stable. They don't last long in the body, and so they're synthesized directly on site, on the site of inflammation. They generate inflammation and pain. If you can inhibit their synthesis, then you don't get as much inflammation or pain. And that's why you take ibuprofen or Tylenol or aspirin for fever, for pain. Now, some of the NSAIDs, like aspirin and ibuprofen, inactivate both COX-1 and COX-2. And it was discovered that some of the blocking both COX-1 and COX-2 generated some problems like increased gastric secretion. So if some prostaglandins lower the amount of stomach acid that you're generating, then if you blocked the synthesis of those prostaglandins, what would you expect to happen? Ulcers or indigestion or other kinds of things, right? And people will still tell you, don't take too much ibuprofen, you don't want stomach acid problems, right? So when Professor Simmons discovered COX-2, a bunch of people got to work making molecules that would bind and inhibit COX-2, but not COX-1. Both Pfizer and Merck had some drugs that did this. The trade names were Vioxx and Celebrex and Bextra, and the structures of these are shown in your text. It turns out by inhibiting COX-2, these drugs were able to manage pain and inflammation without an increase in gastric secretions. And so that was, they were touted especially for patients with arthritis who needed to be taking these things constantly. However, both Vioxx and Celebrex have now been withdrawn from the market because they came with an increased risk of heart attack and stroke, possibly because they disrupted the balance between the other prostaglandins that both promote and inhibit platelet aggregation. So that's an interesting story of, and in some ways discovery that discovery that enzyme started here at BYU. Drug discovery was done elsewhere, but that's an important example of the interplay of biochemistry and organic chemistry and biology. Okay, so therefore what? What do I want you to be able to do with this? Again, recognize the differences between the molecules, be able to tell a leukotriene from a prostaglandin, from a thromboxane, from a prostacyclin. When it comes to their effects, I don't know that it's necessary to memorize everything. The standard is you want it to appear as though you were in class today paying some kind of attention, all right? You don't need to have a ton of stuff memorized, but you do want it to look like you got something out of class today. Okay, so they wanted me during the first 9 o'clock hour to be explicit about this, and then I realized that what they meant was to be clear and not explicit, so sorry about, no, I didn't. There was no cussing during the 9 o'clock hour. What they wanted me to do was end of test 3 material. So there we'll do that. Now, the remainder of the chapter will probably need 2 days on this. So both Wednesday and probably Friday is terpenes. Terpenes are a class, they're called lipids because they're mostly nonpolar, but they're found all over in biology, from essential oils to cholesterol to some of the vitamins that we talked about today. And their synthesis is beautiful beautiful, simple, and then leading to complicated molecules. So the fun part of this is going to be looking at a complicated molecule and then breaking it down in your mind into its subunits that were used to make the molecule, and then to generate a pretty reasonable hypothesis as to how that molecule formed. And it's going to be based on simple reactions that you already know a lot about. SN1, E1, and addition of electrophiles to alkenes. So before we dive into that, we need to look at the precursors to this interesting class of molecules. And we'll learn about the basic chemistry that generates these molecules. The precursors to terpenes are 5-carbon, what I'm going to call monomers. Well, probably won't call 5-carbon, what I will call isoprene units. So isoprene is a hydrocarbon that looks like this. It's got 5 carbons in it, 4 in a chain with the branching methyl group, and 2 double bonds. You're going to see that if we ignore the double bonds, all of the terpenes share this same basic core structure of 5 carbons, which I'm going to number in a way that makes sense to me. 1, 2, 3 is the branching methyl group, then 4 and 5. If you don't like it, I don't care. You can number it however you want, but that's how we'll talk about it in class. Biologically, the precursors to terpenes are 5 carbon subunits that have a leaving group on them. So you can see here 1, 2, 3, 4, 5, and then on carbon 5, we're going to have a pyrophosphate leaving group. In this particular terpene, there's a pi bond between carbons 2 and 4. This one is called dimethyl allyl pyrophosphate. It's a big mouthful. Here's the dimethyls, and then the allyl refers to the fact that the pyrophosphate leaving group is on the allylic carbon relative to that double bond. So, leaving group. Couple of things about that pyrophosphate leaving group. Probably it involves magnesium as a counterion to neutralize some of the negative charge on the phosphate oxygens. The other thing to recognize about pyrophosphate is that it has a phosphoanhydride bond, and we've learned about anhydrides before, and we've said that phosphoanhydrides are about the same reactivity as anhydrides. That is, they can be attacked by nucleophiles to generate an intermediate where then electrons kick down and a leaving group leaves. One of the reactions that can happen to pyrophosphate is a hydrolysis reaction in which water attacks one of the phosphoruses, and the byproduct is 2 molecules of phosphate. And this is very downhill in energy. It's very exothermic. And your biochemistry texts will call this phosphate ion inorganic phosphate, and they'll abbreviate it often as a little circle with a P and a subscript I, where the I stands for inorganic. Why do they call it inorganic? Because it's not attached to carbon. The fact that pyrophosphate itself, after it leaves, can undergo a hydrolysis reaction that's very energetically favorable makes this a super great leaving group, because not only does it leave, but then it splits apart into something else, and that helps drive the reaction forward by Le Chatelier's principle. So you'll see that happen a lot in biochemistry with the pyrophosphate as a leaving group. You'll also see your biochemistry texts abbreviate that pyrophosphate group as O, 2 P's, and an I, and that stands for when it leaves, it's pyrophosphate, and it can be split up into 2 inorganic phosphates. The other precursor has the exact same carbon skeleton, but the double bond is instead between carbons 1 and 2, instead of between 2 and 4. And we're gonna see next time how this difference allows for interesting chemistry that allows us to combine these two precursors into longer chains. All right, with that, good luck on the exam. I will see you Wednesday.